everyone, and welcome to the Micro E-Learning program from the Global Parkinson's Genetics Program in Aligning Science Across Parkinson's. My name is Ruth Schneider, and I am a neurologist and movement disorder specialist at the University of Rochester in Rochester, New York. Today, I am going to discuss how to identify and phenotype at-risk carriers of Parkinson's disease-associated genetic variants in decentralized studies. I will start by reviewing methods of identifying non-manifest carriers, those genetically at risk for, but without a clinical diagnosis of Parkinson's disease, then provide a comparison of traditional versus decentralized study designs, review methods and types of remote assessments, discuss tips for improving study retention in decentralized studies, and lastly, review how to handle risk disclosure in new diagnoses. There are three main ways to identify genetically at-risk carriers that do not involve widespread genetic screening: use of existing research cohorts, collaboration with direct-to-consumer genetic testing companies, and targeted clinic-based testing. In some cases, researchers may be able to take advantage of existing research cohorts of genetically at-risk carriers, or cohorts of Parkinson's disease programs where outreach to family members may be possible. Direct-to-consumer genetic testing companies have broadened the landscape of genetic testing and vastly expanded the pool of people who are aware of their genetic status, providing a new method of reaching potential participants. Lastly, increasingly, we may see people who have undergone or are interested in undergoing genetic testing in the clinic to understand their personal risk of Parkinson's disease in the setting of, for example, a strong family history of Parkinson's disease. While this is not common practice now, it is likely to become more common over time as the science of prevention advances. Regardless of the method of identification used, recruiting a sufficiently large sample of non-manifest carriers using a traditional in-person study design is likely to present significant logistical challenges. Such a design would require multiple sites, substantial infrastructure, and large numbers of personnel. In this case, participation would be limited to those living within a reasonable distance from the selected study sites or willing and able to travel. Decentralized study designs, which enable the collection of data from participants in their own environment, provide an alternative model. Decentralized studies can be conducted from a single site, require relatively little infrastructure, and need fewer personnel. However, while decentralized studies reduce the impact of geography on participation and enable more frequent assessments in the real world, they are not without drawbacks. For example, they are often heavily reliant on technology and therefore risk generating new barriers to participation. Assessments can be more difficult to standardize, and some assessments, such as lumbar puncture and neuroimaging, are simply not possible in the home environment. That said, for the purpose of phenotyping genetically at-risk individuals, I think there are more advantages than disadvantages, and we need not take an all-or-none approach. Hybrid studies with in-person and decentralized elements are increasingly popular. Decentralized studies may incorporate the remote capture of participant-reported outcomes and clinician-reported observations, the use of digital health technologies, and biospecimen collection. Patient-reported outcomes, or PROs, can be easily administered asynchronously via the survey functionality of many electronic data capture systems. Clinician-reported observations can be captured during synchronous video visits using audio-visual conferencing software, but may require some modification for remote assessment. Here is a by no means exhaustive list of relevant motor and non-motor assessments that can be incorporated into decentralized studies. Decentralized studies may also take advantage of a growing number of digital health technologies, including smartphones, wearable sensors, fitness trackers, ambient sensors, keyboard-based tests, and computer vision for the objective in-home measurement. Each type of digital health technology has different advantages and disadvantages, and what exactly can be measured differs from product to product. Broadly speaking, digital health technologies can be used to capture vital parameters such as blood pressure, heart rate, physical activity metrics such as step count and time spent in moderate to vigorous physical activity, cognitive performance such as information processing speed, and motor signs such as bradykinesia, tremor, and gait impairment. Lastly, we should not forget about postal mail. Participants can be sent smell tests, biospecimen collection kits, and environmental sampling kits for use at home and subsequent return. When these methods do not suffice, researchers can partner with other organizations, such as mobile phlebotomy vendors, to augment the study design. With longitudinal decentralized studies, we do need to be particularly mindful of maintaining participant engagement. A successful multi-pronged strategy is likely to include many familiar tried and true methods: easy scheduling, friendly and personable communication, reasonable compensation, and tokens of appreciation. Digital newsletters, online webinars, and participant-oriented study websites can also be useful tools. Lastly, do not underestimate the value and importance of a thoughtful study design. While as researchers we often want to collect as much data as possible, we must carefully consider and balance this against participant burden. Early and ongoing involvement of individuals with lived experience can help inform study design and retention strategies. Lastly, when working with genetically at-risk individuals, we must be mindful of how we handle risk disclosure in new diagnoses. Particularly at the start of their study participation, participants may not be aware of or have fully digested the health risks associated with their genetic status. Secondly, it is possible that participation in a clinical research study may result in a new diagnosis. For example, examination findings concerning for Parkinson's disease may prompt a referral back to their care team and a subsequent diagnosis. Information regarding risk and the potential for new diagnoses should be clearly described during the consent process, and research teams should provide resources and avenues for more information and support when desired. Including individuals with lived experience in the design and leadership of research studies can provide an often missing perspective and help guide decisions regarding risk and result disclosure. In conclusion, 1, we need multiple avenues to identify genetically at-risk individuals. 2, decentralized study designs may make it easier to phenotype genetically at-risk individuals at scale, though there are some trade-offs. And 3, with this population, we must be mindful of maintaining study engagement, managing risk disclosure, and managing new diagnoses. Thank you.